Right, our next speaker is Fabio Fiorani from the Forschungszentrum Jülich, and he has been for many years the head of the Jülich Plant Phenotyping Center and has there taken responsibility for the development and implementation of lots of new techniques and platforms and approaches. And today he will tell us an example of the application of the platforms on analysis of phenotypic variation in morphophysiological traits and seeds, secondary metabolites in Chenopodium quinoa Chilean ecotypes. So,
yes,
Fabio,
yes, thank you. Thank you, Thomas. Yes, so the presentation will not really be focused on platforms, but is a project-specific and crop-specific presentation. However, I make some consideration that fit well in the theme of the workshop and also present a new platform at the end of my talk. So yes, QUINOA, this was a project funded by the German Federal Ministry of Research and it's a collaboration with the University of Aachen and the Institute of Agriculture of Chile. So quinoa is actually a species belonging to the Chenopodiaceae family and it's called Chenopodium quinoa, originating from the Andean region. It is adapted to harsh environmental conditions, and in this, in the existing variation there, it's interesting also for future climate scenarios and also for different cultivation in different latitudes, as I will mention during the talk. What is very interesting is that quinoa produces nutritionally balanced seeds, and a reference genome has been sequenced a few years ago, and there's a way more ongoing on resequencing projects. And together with the University of Aachen and Björn Zadel, we have done some resequencing also in this project. But still, obviously, it's compared with many other crops. Both phenotypic and genetic diversity remains largely unexplored. However, as the picture on the right shows, there are many quinoa products that have reached also European markets and they are popular in South America as well. Okay, so in this project, quinoa, as I said, originates from the Andean region. It's one of the center of origin. And the current germplasm has different biomes, as indicated here by the color dots. And in this project, we focused on the coastal lowland biome and selected over about 110 ecotypes that belong to this biome. One of the reasons I mention it right now, one of the reasons is that the previous research and some other genetic considerations showed that there is most of these ecotypes should be day neutral. Therefore, the photoperiodic requirement should also be good in the sense that it can be cultivated at different latitudes. Indeed, flowering time, we checked for all the genotypes of this collection, the flowering time in greenhouse in Jülich under our greenhouse conditions. And here the graph shows days to flower on the x-axis and the coded quinoa genotypes on the y. In short, there is almost a threefold variation, at least in this experiment in our conditions, ranging from less than 30 days to more than 70 days to flower. We collected also other data such as plant height and days maturity. We looked even at seed productivity and thousand kernel weight, as well as panicle shape. And this is a PCA analysis showing how the load of these components on the PCA axis, showing again that is the first indication that in this material and in this collection, there is a sufficient variation in the simple traits, showing also that there are subgroups that can be classified according to these traits. So that was good news. Now, one thing that is very important looking at potential future exploitation of new QUINOA germplasm and going to pre-breeding and then breeding is the content of saponins, saponin in the seed pericarp. Saponins are triterpenoid substances that confer also a bitter taste, at least some of them. And traditionally, they need to be removed before quinoa seeds are consumed. So this is costing time and money, of course, and breeding efforts also in Europe, for example, in the Netherlands, have been looking for so-called sweet quinoa genotypes. So we characterize with LC liquid chromatography and mass spectrometry the saponin contents of the seed pericarps in this collection, and we found actually that according to prior classification with a cutoff around 6 milligram per gram content of saponins, that we have only 1/3 of our collection could be classified with high saponins, but we have many genotypes with low or average in this case and low saponin content, which is actually encouraging. We looked further into a few individual sapogenins compounds, and this is what is shown on the right, but I will not go in detail there. Of course, there are not only compounds that need to be removed, but compounds that are beneficial, such as phenolic compounds, where quinoa is supposed to be enriched in. And here it's a clustering of the total bound phenolics and a few individual phenolics that we identified with LC-MS-MS. And here in the highlighted columns in right here, these are total phenolics. Next to the right, the free phenolics. You can see going from red minimum to blue maximum, there are different clusters there. There is variation, as well as in the bound phenolic compounds here on the left, where there are actually a few accessions that have pretty high content of phenolic compounds. So taking that together from a metabolite profiling in the seed pericarp point of view, we have good variation there and could be exploited in the future. Next was to focus on a small panel of these ecotypes, actually 10, that were further selved by our collaborators in Chile. For at least 3 generations, there will still be a certain degree of heterozygosity, but this has been reduced over time. And do a field experiment in Chile in the southern Atacama Desert. You see a picture of our field site with no phenotyping infrastructure whatsoever, and therefore basically pushing us to use portable instruments and use our knowledge of plant physiology to do the right measurements. Our field sites look like this. We had a split plot design with full irrigation and reduced irrigation subplot, and genotype plots that were replicated 3 times. Okay, environmental conditions. So I want to spend a couple of minutes in that because this, I think, is relevant. So it's a desert, and therefore, point number 1 is if you don't give water, plants do not grow. Therefore, when designing an experiment where you are experimenting with water supply, you have to give a full irrigation, and also for the plants that will eventually experience symptoms of water deficiency, you will have to supply irrigation anyway. So here we had a reduced irrigation schedule, as it is shown by the soil water content on the lower panel here. During phenology, we applied then 50% of the maximum or the full irrigation water based on the agronomic experience of our collaborators in Chile in that location. And this was the experiment look like. It doesn't rain, so the average rainfall there is 0. And when you look at temperatures, is actually something that already would give to some older growth chambers some issues, because we have a pretty large differential between day and night temperature, as you can see here. So ranging from, for example, during the day, we have an average of 25-ish over the entire growth season, but in the night it's much lower. High VPD, especially high VPD peaks, and very high irradiance throughout the growth season, I would say. So I remember the one remark of Kerstin Neumann yesterday during our talk. She said, yes, well, field conditions are very difficult to repeat. Well, absolutely right. There are some fields in some specific cases where you can expect that if you do this experiment there this year or next year will be really similar. Okay, what we measured was something that we could do in a very quick way, such as plant height, standard diameter, and then physiological measurements like water potential, relative water content. We did gas change, water use efficiency, chlorophyll fluorescence. We used thermal imaging, spectral imaging, and we also excavated roots and measured yield at the end of the growth cycle. Now, the root excavation, I'm not going to talk about it because the main message there is that there was really no big difference that we could find in root angles, for example, at least in our relatively low replication experiment. So the treatment worked in the sense that this is yield, that's the top panels, and this is seed weight per plant, and FI and RI stands for full irrigation and reduced irrigation. And these are genotypes compared with a commercially available genotype in Chile, which is actually tolerant to reduced water availability. Therefore, we used it as kind of control. The good news is that, well, there are two good news. One good news is that our treatment somehow worked. You see that the green dots show overall that there was a reduction in yield, albeit not really severe, and mirrored by a reduction in seed weight per plant. However, we started then to look at individual genotypes and at their response, because there are clearly genotypes that show minimal responses to the treatment, or actually no responses to the treatment, similar to our, let's call it, control genotype. We clustered then, or looked at PCA analysis. The top panel shows the treatment effects, essentially reiterating what I just said using the previous graph, and then looking at individual traits and their contribution. That's a lower panel, where I will then go specifically in a few of them. Of course, green ones, for example, if we focus on relative water content or on the seed caliber with a threshold of 1.7 millimeter, in this case, plant height, you can see that they have a high contribution. And there is this LTL1, we'll go in more detail now. This is a thermal index that we calculated. And if we look at this data from a correlation matrix point of view, actually our thermal index sits here. And this is measured with cell phone, actually a FLIR microthermal camera. And in fact, you know, the correlation is not spectacularly high, clearly, but there is some correlation with the yield and seed weight per plant, which is something I personally didn't expect necessarily. On the other hand, you have classical easy parameters to measures like plant height, for example, that have very good correlations with biomass, of course, and also stem diameter, and some decent correlation, at least from, depends on the perspective, with some yield parameter. The bad news is that we spent actually a lot of time measuring quantum yield of photosynthesis for all these plots. But even when we split our data set in high light conditions and low light conditions relative, depending on the time of the day essentially, and sun angle, we couldn't really find any correlation with any parameter. Next, we used here on the right side, we used a hyperspectral imaging system, which is a portable handheld camera. And we took many pictures plot by plot and then analyzed the data, as I will go in a minute, just for a little bit more technical specifications. This camera is a SPECIM camera. The spectral range varies from 400 nanometers until 1000 nanometers and has a spectral resolution of 7 nanometers. So what we did here, we looked, this is relatively complex to read, but I will guide you through. What we looked here was a pairwise comparison for all the spectral indices that genotype pairwise comparisons. So for example, first one is CLS3 and our reference variety and their full irrigation, both genotypes. And then in this diagram, we have plotted the, here on the left side, the size of this rectangle means how many of these pairwise comparison are represented in here, and on these right rectangles, the spectral, how many spectral indices then can explain with significance the pairwise comparison differences. So if I take then a conclusion from this rather complex diagram, I would say that the red-green indices, named the two shown at the bottom here, are the ones that have the most explanatory power for the largest number of pairwise contrasts that we set up, whereas other indices seem to have a very good discriminatory power for specific, very specific limited number of pairwise contrasts, such as some that you see here. So in conclusion, I would say that this was also very pleasing to see that simply using this handheld camera, we could get so far as to explore, which was the scope of this experiment, explore genotype by genotype pairwise comparison and the effect of our treatment. So I would like to conclude the project specific part of my presentation by making some conclusion and giving three simple perspectives. So based on our work, there is a good morphophysiological variation in the Chilean coastal quinoa lowland germplasm we explored. We saw that actually portable instruments and imaging techniques show a very good potential for throughput in field phenotyping of quinoa and future studies. The work I presented in the field was done by 4 people, including me, in approximately 5 days, at least for the measurements I presented. And that there is diversity in these germplasms that can be exploited in breeding programs in the future. Actually, based on the field experiment I presented, there was one variety that was a new variety that was registered in Chile, which was a tangible outcome of our contribution. So I talked very briefly about, I said we did root excavation there. Oh yeah, sorry, I forgot that to mention obviously that the resequencing of this genotype was done and still ongoing to some extent. And that thanks to the work of Katrin Dumschot together with Bjorn in Bjorn Zadel group, a few markers are starting to emerge for simple traits like days to maturity, height to mature at maturity. And these are two different methods where the height of the peak may differ in this comparison because of different algorithms and because the SNPs need to be cleaned up and further filtered. So this was the overall bridging scope of the entire project. The consideration I wanted to make is the following. So when you go in the field and you excavate roots, for people that have done that, one challenge is, besides the manual work, is that you will have a lot of variation, and that it's very difficult to pull out, my experience, to pull out significant differences unless you really, really repeat. And therefore, riser boxes in greenhouses are actually extremely good for looking at simple root trait architecture, at least in the dimensions afforded that are afforded by riser boxes. So here I go back in QUINOA. We did experiments years ago, actually, with a Chilean student that was working with us, Jose Correa, and he took some genotypes that he was interested in and looked at them in riser boxes, and showing that in this case, then for primary root would have significant differences, at least between some of these genotypes. And therefore, the scope will be also to include more root phenotyping for QUINOA in the future. To that extent, I wish to close my presentation by showing you our new, meaning new that has come finally online, facility called Cross-Screen Riser 3. As you have learned, these were major developments in the German that were realized within the German plant phenotyping networks, and this facility is then open for access for German partners, as it was shown yesterday by Uli Schur. Okay, so this is a short video. We have 1,000 of these cassettes where we grow where we have 4 riser boxes, and therefore 4 plants, let's say, for one of each cassette. So we have 1,000 riser boxes, not 1,000 cassettes, so total 1,000 capacity. And we move them around with these vehicles that are vehicles that they have autonomous vision, but we use them in a different way because of security. We then transport the riser boxes to imaging stations that allow us to measure them all in one day. We use RGB imaging. Let's stop here for a second. Oops, reload the video, sorry. That's, I jump to the relevant part. So I was at the imaging station. We do RGB imaging, and then of shoot and root. And then, of course, when you go to very large numbers, you need to, your accessory facilities, let's call them in this way, soil preparation, need to be scaled up. So this is our assembly line for riser boxes, where we have a compression tool, and then some facilitated way to screw the plates of the riser boxes to the frame. This facility is online, and we now are populating it with projects. So this is essentially what I wanted to say, and I thank again Thomas for allowing me to look a little bit more specifically at one project that was the one that I presented in QUINOA. Thanks very much.
Thank you very much, Fabio, for this nice presentation. It's great to have an example of a detailed study applied to a particular question, or on a particular question. And the talk is open for discussion, questions. Mark first, and then Kerstin.
Hi, Fabio. Thank you for this talk. I have one critical question towards your environmental parameters. You plot global radiation, and you plot values of like 8 to 9,000 watt per square meter. So if I compare this to the weather station of the IPK, we have something like 800, 900. Yeah, exactly.
Yeah.
And normally the factor to recalculate into micro Einstein is like 2. So it would translate into your values like 16,000 micro Einstein, which is a lot.
So if you, of course, there was not only 9,000. So 9,000 was the end of the scale. So variation is between. I'm looking
5,000, yeah, but still it would translate to 10,000 micro Einstein. So I just wonder how this is measured. So what
well, it was a radiometer, because this was the weather station of the research station in Chile. And yeah, I don't know, we can look again. Radiation is very high, that is for sure. But I would agree, sorry.
It would be like an order of magnitude higher than what we have on sea level. So
I would agree. Yes. So I would agree that maybe we can look into that again. However, you're talking about one of the places on Earth with the highest radiation in general. I'm not saying that this is, you might be right. Maybe there's some issue of calibration and so on. However, you're talking about one of the places on Earth with the highest annual radiation.
I understand that, but still that would imply that you have something.
Yeah,
like
and as I said,
ultraviolet radiation or something which is hitting your sensor there, which is saturating its energy.
Okay.
Okay, but thank you anyhow.
Yeah.
Right, Kerstin.
Yeah, thank you, Fabio, for this interesting talk about a controlled field environment, more or less. So I'm not sure if I fully understood. For me, the impact of drought on the yield seemed to be rather low. Of course, there were genotypic differences, but the overall reduction didn't seem to be very high. So I maybe missed. So in this reduced irrigation, so how strong was the reduction in watering in the drought treatment?
Yeah.
That's the first question. And maybe also the second to the correlation, phenotypic correlation plots. So you didn't have, I think you emphasized that you didn't have a correlation of photosynthesis to the overall yield. So was there a strong reduction in photosynthesis caused by the drought? And also, did you observe an effect on phenology caused by drought? So was there a kind of drought escape mechanism visible?
Right. Okay. So I go one by one into your questions. So how did we set up this experiment? So as I said, there is no rain. Therefore, you have to irrigate plants. So the full irrigation was set up. So for during the vegetative stage, essentially, we had the same irrigation. Then we started with a reduced irrigation schedule was 50%. Of the total amount that would be delivered normally when you have the selection plots, so that's a full irrigation treatment. We couldn't do this in a more clever way because we wanted to use a evapotranspiration model to really meet crop demands, but we realized that the crop coefficients that you would need to recalculate them, and it wasn't really possible due to resources in the project, so it means more measurements, and we couldn't do that. So we relied on the experience of growing quinoa in those plots in that location by our Chilean partners. The treatment was not very severe, but the average reduction was about across all genotypes about 25%. Therefore, we could have gone a little bit lower, but then it becomes really risky because in that environment, you cannot really access water reserves deep into the soil. The other part, I think I have addressed your first two questions, Kerstin.
Yes, the correlations, yes. Exactly.
And then the correlation,
how strong was exactly? Yes, thanks.
So in fact, when I go again into, and I'm looking at this matrix, you can still see my screen, right?
Yes.
Yeah, so I make it bigger. So in fact, you can see that if we take PS2 efficiency, low light, highlighted, these were the two traits that we looked at. We split it at the data set in low light, highlight, relatively speaking. Actually, it doesn't correlate with anything. That's the thing. One point could be that, of course, we had a rather low replication again. So we had 3 plots per genotype. But you would expect to have some correlation with biomass, but there wasn't. Yeah, that's what we found.
Just a short question. So how strong was photosynthesis reduced by the drought in comparison?
Overall, if I look at the average, average reduction was again 10–15%. So yield more strongly, yeah, than photosynthesis.
Thank you.
Yeah.
There's a question by Ben and then a question by Hendrik.
Thanks, Fabio. I'm not sure if I understood correctly. So I think you mentioned at the end that you did root phenotyping in riser boxes for QUINOA, and you also mentioned that you did some phenotyping under field conditions as well.
Mm-hmm.
Would you be able to make a comment about similarities or dissimilarities which you see in the root phenotypes between the two?
So what I didn't explain well was that the phenotyping that I showed was done years ago and on different genotypes. Therefore, we cannot correlate them directly. However, we have done also, and we are looking at the data for the same subset that we used in the field. We also done root phenotyping in the greenhouse, but we don't have data yet. It is very difficult to correlate. So first of all, in the field, I couldn't see any difference. So nothing statistically significant. Therefore, that's already not that great, to be honest. And therefore, the experiment that it has been done in the greenhouse in riser boxes will be very difficult to correlate to the field one. There are instances in which you can do this, also in other crops. And so here in the field, I've been using, let's say, a shovelomics method and a goniometer to look at root angle and root densities. There was no significant difference between anything, essentially some variation, but nothing that I can talk about.
All right, Hendrik, please.
Fabio, could you go back to your correlation table?
Yeah.
I was not sure how to interpret it. Is this only for the low water treatment where you correlated different genotypes, or is it more complicated?
No, I think, no, this one, no. So this one would be on the, on the effect, not only at the water treatment. Yeah.
So this thermal index that you pointed out, what I'm not sure. There is a small negative correlation. What is the thermal index exactly? And does it mean that in the end, the transpiration is higher for the high yielding lines or is lower for the high yielding lines?
Generally speaking, yes. And the thermal index here, as I said, technically we measured with a cell phone, and the equation that we used and the method that we used was wet leaf temperature minus the ambient temperature. So yes, to answer your question, you can expect to have moderate correlations with transpiration and then yield in this case. As when you look at the strength of the correlation, you are around 0.5. So I mean, it is not extremely high. Yeah.
Thanks. Yeah.
All right. Before we hand over to Rick, Eva, you had raised your hand, but now it's gone again. Do you still have a question or not?
It needs some more thinking. I haven't finished formulating it, so
Okay.
no.
All right, then we hand over first to Rick.
Yeah, Fabio, thanks. I have a question about the same sheet that is now in front of you.
Yeah.
I was also looking at it for a while. I think there are lots of information hidden here because there are kernels that are different in size.
Sure.
And also in orientation. Can
Orientation?
You, for instance, yeah, some are
orientation of the correlation.
Pointing to the left.
Yeah, sure. Yes, I didn't go specifically in all of this. There's more.
But explain only the row of the H plant height. Then this is
yes. So the H for one thing stands for harvest. So these are parameters that were measured at harvest, and these, the I, were parameters that were measured at the inflorescence appearance stage where these physiological measurements were made. So if you're talking about plant height measured at the, so that's not the final plant height, but it is plant height at flowering, let's say. It correlates rather well, actually pretty well, with stem biomass and with leaf biomass and with stem diameter. It also correlates to some extent with the thermal index I've been talking about, and it correlates with itself at harvest and with yield in a positive fashion. Although, you know, this color scale gradient, where you can see that we could argue whether a correlation of 0.5 is good or bad, depending on the aim, I would say. But yes, so this is plant height. Did you have other specific questions, Rick?
That takes too long. But
you can ask me later. If you're,
then the 10 rows lower, there is H plant height, and then some are blue and some are red.
Okay. Yeah, so what you can see here, the red ones, so the blue ones are with yield expressed as seed weight per plant, or 1,000 seed weight, with somehow with the panicle shape, if you say length and width here, but again, you're talking about low correlation coefficient, so I wouldn't make a very big story out of it. And negative correlations with the seeds that are passed through a mesh of 1.7 millimeter, 1.4 millimeter. These are a little bit more difficult to explain, obviously. One could say, well, essentially, would I expect any correlation there? Not necessarily. Therefore, I can't really say much about this.
Much clearer now, thanks.
Okay.
All right. Thank you very much. Well, unless there is another urgent question, I think we should stop here and thank Fabio once more for the presentation and for the discussion.